So this describes the structure of skeletal muscle, or is a depiction of the structure of skeletal muscle. At least longitudinally, like we typically observe it, or like you would imagine, looking at it from the side. And today we're going to spend a lot of time discussing how this changes with contraction. So this is not a static representation. These bands change with contraction. But these are the usual suspects, and you can pick out a few other organelles that I won't go into in detail, but the proteins, the bands, everything as would be arranged within skeletal muscle looking at it from the side. It looks a little bit different if you look at it from the end. These are myofibrils cut cross-sectionally, so looking at it end on, and you can see the order. It's really impressive. The degree of order of the actin and myosin filaments within the myofibrils. Myosin, of course, the larger of the dots that are here, the little small circles, and they're surrounded on all sides by actin, which will become important as we think about shortening and contraction. I often show you these in 2 dimensions only, and you see one-on-one filaments. But that's not the case. The myosin strands are surrounded by actin, and the heads of the myosin molecules point in all directions to take advantage of that. It allows a lot of surface area, a lot of contact between those filaments, which allows a lot of tension generation and a lot of force and a lot of shortening, which we'll talk about today. So we know what skeletal muscle looks like. And you could probably even describe this entire picture, or most of this entire picture to me, now. Some of the elements within the muscle you won't be able to explain right away, but you will by the end of class today. So you spent a lot of time learning about the structure, then the signal and the neuromuscular junction, and now the movement, the squeezing, the sliding is what we'll talk about today. I need to get my money's worth from these animations. So I'm going to show you a quick video again just to take up and review what we know from last week. Maybe. Aren't those smooth? That's some high quality rendering. Okay, so we don't have volume today. Let's not do this. Volume's there, it's just not going through. So I won't then have you review the signal sent at the neuromuscular junction, how it releases calcium from the terminal cisterns of the sarcoplasmic reticulum. I assume that you know that happens. Calcium's in play. And we're moving on from there. What I want you to know, before we get too far, is a point or two about myosin, because myosin is kind of the star of the contraction show. Myosin is what it's all about. Everything else is in support. Myosin does the moving. Myosin does the contraction. And the important characteristics as far as contraction and shortening are concerned are listed here. There are more aspects or characteristics of myosin. None of these are part of the tail of myosin that makes up the stalk or makes up the filament. But the important parts of the myosin head are listed here. It has a site that binds ATP. And I'm assuming, we haven't spent a lot of time talking about ATP, but you know that's the energy currency of the cell. That's what we use to do work. We break it down to harvest energy to do work in the cell. Myosin can do that. It binds ATP. It also does the job of breaking it down. Myosin has an enzyme in the head that breaks one of the phosphates off of ATP. You're all familiar with ATP, correct? Adenosine with 3 phosphates. Break one of them off and we can harvest the energy from that bond. Myosin does that. The other important aspect is it binds actin. A third separate site allows it to connect to actin and make a cross bridge. These are the elements that we're going to order and discuss today to understand contraction. And contraction goes according to the sliding filament mechanism or the sliding filament theory. So after depolarization, after the neuromuscular junction, if excitation-contraction coupling exists, the sliding filament theory occurs. The sliding filament mechanism occurs. And all that describes is that myosin walks up the actin filaments. And that's what you're seeing here in this animation. Myosin's walking up the actin filament. Now obviously this is an over-exaggeration. This is going on indefinitely, and it can't do that. You can't indefinitely contract muscle. And we'll go into the ingredients and details of this picture, but what I want you to see is that sliding aspect. Myosin and actin going in different directions. Myosin walking up the actin molecule. So of course this redhead is myosin, this filament with all its pieces is actin. What that means is the sarcomere shortens. The bounds of the sarcomere we said were the Z discs on the end. If you measured a sarcomere at rest and you measured a sarcomere in contraction, the distance between the Z discs is smaller. The sarcomere is smaller. So that's a large part of contraction. And it doesn't happen by compressing or expanding the proteins, or compressing or expanding the filaments. They stay fairly rigid. And we talked about the structural proteins that are in play to maintain rigidity, the structural integrity of the muscle. They ensure that as myosin walks up the actin filament, that force, that pull, is transmitted the entire length of the muscle. The filaments don't expand and contract like an accordion. That's how we initially thought muscle contraction worked. We thought muscles were an accordion that inflated and deflated. And then when we got more advanced technology and could do muscle biopsies to look in at the muscle and get a sense of what's actually happening, we realized it's not expanding and contraction. It's not, expanding and compressing. It's not an accordion. So these filaments slide on top of each other. The sarcomere shortens, the filaments stay the same length. What changes is their degree of overlap. When contraction occurs, when calcium is present, and the muscle, the sarcomere shortens, these filaments overlap more. When the muscle is at rest, when you're not contracting and it returns to normal, it elongates. The filaments overlap less. So in a resting situation, in the top band, as you can see here, some small degree of overlap. You have obvious H zone and A bands, which are the locations where there is only one filament present, no overlap. I'll show you those here. The width of those is a certain amount at rest. And then as the muscle shortens, And these are progressive figures, so it's partially shortened halfway through the range of motion and then fully contracted at the bottom. I'll overlay the original widths. These are the same rectangles, copy and pasted, and then compare them to the new size of the I band and the H zone. So those zones with only one filament present seem to have shrunk. And the zones have shrunk. Less of the sarcomere is taken up by only one type of filament. There's a lot more overlap. Compare those two situations. So the distance between the Z band shortens, the sarcomere shortens. But sarcomeres are really small. You can see it with the naked eye. You can see the striations. But one sarcomere shortening does not a muscle contraction make. All of the sarcomeres in line shortening together is what defines contraction. That's what allows the muscle to shorten. So it's all of those happening in tandem that allows the muscle to move through a wide range of motion and the joint angle to close. So let's consider how that happens. And I think this slide is not one that's in the notes, so you might have to take a few more notes. So let's think of what occurs. Let's go through in detail this diagram. And Matt would have left off in class talking about calcium. That's the tangible end result of depolarizing the muscle. Depolarizing the muscle and the release of calcium from the sarcoplasmic reticulum. Last step in depolarization, first step in contraction. You could call that the link between excitation and contraction coupling. So calcium binds to one of those regulatory proteins. I was going to say support, but one of those regulatory proteins we talked about last week called troponin. Troponin is shown in this animation as the three purple balls in a protein attached together. And when calcium binds, it undergoes a conformational change. You'll learn all about conformational changes in biochemistry next year. Really, when anything binds, the shape changes. And calcium binding to troponin is no different. The shape changes inside the cell. And that causes it to pull on these blue filaments called tropomyosin and pull them out of the way. What are they in the way of? They're in the way of the binding site. They're preventing myosin from binding to actin. But as soon as calcium binds, and troponin undergoes that conformational change, and it rolls tropomyosin out of the way, the path is clear for myosin to bind. So myosin binds. Myosin binds, and at this point, I'm going to assume that we have ATP present. We'll come full circle, but myosin binds with ATP and then breaks down that ATP, hydrolyzes that ATP. Hydrolyzes is a fancy chemistry word for breaks apart with water. Hydrolyzes. And so if ATP has 3 phosphates and we hydrolyze it, we remove one phosphate, we're left with ADP, adenosine diphosphate, and an inorganic phosphate. I know I'm bordering on biochemistry, but it's important to understand this. Doing that gives myosin energy. It energizes the myosin protein. And we use that energy to accomplish work. The work we're accomplishing is shortening. And that shortening is referred to as the power stroke of contraction, or the power stroke of this myosin cycle. the energized protein can then pull on the actin molecule and shorten the sarcomere. ATP is hydrolyzed. ADP and phosphate are still bound to the energized myosin, and it's the returning to normal that causes the power stroke. Returning to the normal shape that causes the power stroke. I suppose one thing that I missed that would help that make sense is that when ATP binds, myosin reaches, myosin extends, and then it's the returning to normal that defines the power stroke. Now, what happens with ADP and phosphate is still up for debate. Power stroke occurs with them intact, or does one of them detach, or do they both detach? We're not sure. As recently as this year, we're not sure, and I'll show you that in a second. Eventually, they both detach. That's part of the power stroke. They both have to detach. They leave the ATP binding site because we need to make space for the next ATP to bind. And it's only when ATP binds that myosin lets go. Myosin is stuck, attached to actin, until it binds a new ATP molecule. When it binds, it lets go. When it binds, it reaches, and it starts the whole process over again. If calcium is present, it will bind to actin. It will break down ATP. It will create the power stroke, cock the head, and then walk up the actin filament. Of course, if calcium's not present, it can't bind, so this doesn't happen. Calcium is permissive in the sliding filament mechanism. So the connection, myosin on actin, is what we call the cross bridge. Myosin attaching or binding to actin is called the cross bridge, and that cross bridge breaks when ATP binds and myosin releases. And the only way we can have contraction is when those two are in contact, when cross bridges form. Incidentally, when you die, you run out of ATP, and the sarcoplasmic reticulum becomes very leaky. So that point where myosin is attached to actin and it needs ATP to bind to let go, that's called the rigor conformation. You've heard of rigor mortis, rigor. When you die and you're not making ATP, myosin can't detach. All the sites are open because calcium has leaked out. All the myosin binds, and muscles are rigid. The entire body becomes rigid. That's why. We're going to talk about metabolism later this week, but you probably only have about 3 to 5 seconds of ATP in your muscle at any one time. You're this close to rigor, sitting there. Shocking, isn't it? 3 to 5 seconds, that's it. So we better have a way to replace that ATP. That's getting ahead of myself. So all of those steps you see listed in a diagram like this in your book, and they all generally agree. There are a couple points that differ and differentiate between the two, but they generally agree. So myosin hydrolyzes ATP and it reaches. You see that here. ATP is bound. When it hydrolyzes or breaks them apart, the head reaches forward. See how it's angled a little bit up and to the left? When you hydrolyze ATP, it reaches, it's pointing straight up. And that's what the angle is trying to describe. So you see the angle shown here. I've highlighted it with this blue, which I guess kind of helps. But it's describing the angle between the head and the filament. It's not describing the angle of this neck of the myosin molecule. It's the head and the filament. So it reaches to 90 degrees. The ADP and the phosphate detach. This is one of the points where the book doesn't agree. The book says phosphate comes off first. Maybe. Maybe they both come off. We're not sure. They both come off at some point. And the power stroke returns the angled myosin head to normal. It returns it back to 45 degrees. That's what characterizes the power stroke and allows the sarcomere to shorten. Break up the phosphate from ADP. Myosin's energized. One or both of them detach, and the molecule returns to normal. Then to complete the whole process over again, myosin has to bind ATP to detach, and then the whole cycle starts again. It reaches, hydrolyzes, shortens, detaches. 4 major phases of the sliding filament mechanism. So that should be fairly clear. Really, this diagram is self-explanatory. What I wanted to show you was the angles and what the angles referred to, and that it agrees with the animation on the last slide. One fun exercise for you will be to review the video and see the timing in that animation. Does ATP bind first? When does it hydrolyze? How does calcium bind? When does it open? When does it reach? See if you can decipher all the ingredients that we've listed here. And they don't all agree. So we can trust the book that phosphate detaches first. This is what I was referring to when I said even as recently as this year we don't know what happens. This is a paper from 2018 that's still trying to decipher the myosin-actin interaction. And this is a simple cartoon diagram of the process. There are many more complicated diagrams if you want to look up Sugi et al. 2018. But this describes just what we talked about on the last 2 slides. All the same procedures are evident here. You can see myosin breaking down ATP into ADP and phosphate. You can see the head reaching. You can see myosin detaching with ATP. The difference here is that these both come off together. I'm inclined to believe these researchers rather than the book, as I expect they're professionals in their field. Maybe, maybe not. So, sliding filament mechanism. The muscle doesn't expand and compress. It's not an accordion. The filaments slide over top of each other. The sarcomeres do shorten. The length of the muscle becomes shorter, or it decreases. But the cells, or the action is different than originally thought. Does this make sense? So, you want to contract your muscle. You want to lift your arm up. You want to contract your biceps right now. You think, okay, I'm going to lift up my pen or text someone on my phone perhaps. What is the first thing that happens to engage that muscle, to engage the biceps? Who wants to take a stab at it? Matt? Awesome. Nice. He was on a roll. That's good. That's so many steps. There's a signal that comes from the motor nerves that arrives at the neuromuscular junction. Acetylcholine is released and opens voltage-gated sodium channels. Sodium rushes in. The whole depolarization cycle, very similar to what you've already learned. It's just nerve and muscle, not nerve and nerve. Something happens, calcium's released, and we are at the point where contraction can occur. So what's the first step in contraction? Calcium is released as a result of the action potential depolarizing the muscle. Olivia? Calcium binds to troponin, moves it out of the way, conformational change. What is the result of that binding and that conformational change? Anybody over here? Calcium binds. Troponin moves. Matt, go ahead. Tropomyosin moves. The binding site is revealed. Good. This is going to make an exciting video, won't it? Binding site is revealed. What happens next? I'm going to give someone else a chance. Let's look over here. Who's looking away the most? Binding site's revealed. It's actually really easy. What happens? Want to give it a shot? Yeah. Myosin binds. Fantastic. Myosin, if it has ATP present and bound, will bind to actin. The cross bridge forms. Okay, we don't just want a cross bridge. We want something to move. We want something to happen. What happens next? What does the myosin do next? That looks like a hand race. Myosin binds to actin. It has ATP. What happens next? Yes. Hydrolyzes ATP. Breaks a phosphate off. The myosin is energized. With that energy, what happens? With the energy, what does myosin do? Okay, how about you behind him? Yeah. Myosin takes that energy and does something. Yeah. Myosin head cocks, it contracts, pulls the actin filament. That's what we use the energy for. Returning to its normal resting state. Both of them detach, ADP and phosphate. ATP binds, myosin releases, the whole process starts again if calcium is present and troponin and tropomyosin reveal the myosin binding site. Good. So, the sarcomere shortens. Multiple sarcomeres shorten together and the muscle shortens. But the sarcomere can only shorten so much. The animation you saw was myosin crawling up actin indefinitely, which is not possible. There are limits. And for that matter, there are limits to how much it can elongate and stretch. And that is described by this figure here, which is the length tension curve here for a sarcomere. The length tension curve is a property of muscle fibers. It's a property of muscles as a whole. It's a property of individual sarcomeres. And it describes how much force can be generated for a degree of overlap in the sarcomere. Let's put it that way. Or the length of the sarcomere. So in a resting state or a stretched state, there's very little overlap. Somewhere in the middle, there's some overlap, and near the end, there's full overlap. And the amount of overlap generally correlates with the force that can be produced. So the force of a muscle contraction depends on the length of the sarcomeres prior to and during contraction. The force can change throughout contraction. And this figure on the right shows exactly that. This is a measure of force. Tension in the muscle registers as force when you record it externally. You can see that for a given length, force changes. If the fiber is really long, force is low. If the fiber is really short, force is low. And somewhere in the middle, force is high. The length tension curve of a sarcomere. And this is very similar to the length tension curve you'll see in a muscle or an individual fiber. They both exhibit the characteristics shown here. Less force, very little force at the extremes. And a lot of force generated in the middle. Why is that? Why can you generate a lot of force in the middle? The amount of tension you can generate, first and foremost, depends on the amount of overlap. The number of cross bridges that can be created. One myosin molecule exerts a certain small amount of force. Two of them will create twice that amount of force. 10 of them create 10 times the amount of force. The more cross bridges you can engage, the more force you can generate. And so if you think back to the slide where I showed you rest, intermediate, and full overlap, if there's very little overlap and the filaments are only touching, very few cross bridges can be made. Very little force at the far right-hand side with stretched sarcomeres. As the filaments overlap more and more cross bridges form, more power, more force can be generated. Why does it go down at the end? Because presumably, if you're fully overlapped, the largest number of cross bridges can be formed, the most force can be generated. So the shortest muscle, the shortest sarcomere length, should theoretically have the highest force potential or tension potential. It runs out of room. As myosin walks up the actin filaments, it runs out of room. It butts up right against the Z discs, or close enough that it can't continue to contract. And so even though you have cross bridges that can form, the power stroke can't occur. Myosin can't slide. So when there's too much overlap, also very little tension. Somewhere in the middle, there's an optimum tension. And there's not one fixed spot where it's optimum. It's not always 50% of the way. Sometimes it depends on the orientation of the muscle and the fibers within the muscle, but somewhere in the middle, there's an optimum overlap, where you have lots of potential to make cross bridges and lots of space to shorten. And here, you're seeing this as, this is percent of optimum, so optimum is right at 100. The most force that the sarcomere would generate is slightly longer than the optimal length through slightly shorter than the optimal length. There's a range where force is optimum. And you can apply this not only to the sarcomere, but to the fiber, if you have one fiber, to the entire muscle, if you take out a bicep and then connect the two tendons to force gauges. somewhere in the middle is the optimal force. And they'll generally all look like this. They might look somewhat different, but they'll always have this bell-shaped curve. Low on the ends, high in the middle. So we're going to pick up, excuse me, we're going to pick up with this in a second. But I want to introduce, I think it's worth introducing a concept to you earlier as we talk about length and tension. Because not all length tension curves, not all muscles produce force equally. And so I want you to have this in mind as we talk about maintenance of tension, generation of force, and the other characteristics of muscle as we review what you did in the lab last week. But not all muscles, not all fibers are created equally. And this also helps to understand the organization of motor units as well. We're going to talk about motor units, we're going to talk about recruitment, but I think you have to understand that there are different kinds of muscle fibers, not just one uniform kind. They all contract, but they contract differently. So skeletal muscle fiber types range through two main types and one that overlaps. Slow oxidative, fast glycolytic, and fast oxidative glycolytic. The last one is a blend of the two extremes. You might have heard of these as type 1 and type 2 muscle. Type 1 muscle is slow oxidative muscle. Type 2 is fast glycolytic muscle. And then the muscles that have the best of both worlds are in the middle. We'll call those type 2X, but we'll call them a blend for now. Why are there different muscles? What differentiates these fibers? Slow oxidative, fast glycolytic. It's really in the name. And it will become more obvious as we get into metabolism, which will at this point probably be end of class Thursday, early Friday. First and foremost, the ability to make ATP, the ability to make energy, differs widely between these muscles. And slow oxidative are the kind that are responsible for long endurance type activities. Exercising for a long period of time. They have a lot of mitochondria. They have a lot of mitochondria. They use a lot of oxygen. They can make a lot of ATP aerobically. So one of the defining characteristics is shown here. Lots of mitochondria in slow type 1 fibers. Very few in fast glycolytic fibers. And to go along with that, the mitochondria use oxygen. To go along with that, I also want to point out this myoglobin line just on top. Type 1s have a lot of myoglobin. Type 2s don't. And the reason that I bring that up is if you looked at a muscle fiber, you could differentiate really deep red fibers from pinkish white fibers by looking at them. You can see that in a fiber with a microscope. The color is different, and it's largely due to myoglobin in the muscle. Myoglobin is like hemoglobin. It's very red, dark blue, depending on whether it's oxygenated. But you can see a difference in color between the fibers. So you could pick them out at face value. For our purposes, that's what's being shown here. See these really dark fibers? That's a slow oxidative type 1. Lots of myoglobin, lots of mitochondria, whereas this fast glycolytic, very pale and white, very little myoglobin, very little mitochondria, so it looks whitish or pinkish. So you can see a color difference, and I won't tell you the color difference here is not due to myoglobin because that's going to confuse you. But you can see a difference by the naked eye. Another really important characteristic has to do with the elements or the important characteristics of myosin that I brought up at the start of class. Myosin has an actin binding site, an ATP binding site, and ATPase activity. It's an enzyme. Fast glycolytic enzymes work differently than slow oxidative enzymes. The myosin's different. The protein is different. So fast glycolytic type 2 muscle, the ATP breakdown happens really quick, hundreds of times faster than in slow muscle. Really quick. On top of that, the power stroke, multiple times stronger. Really fast ATP breakdown, really quick shortening. So they're powerful muscles. Powerful. They can generate a lot of force. Slow oxidative have a different type of myosin. ATP is broken down more slowly. The power stroke is slower. And the force that they generate is slower as well, or less as well. They're not powerful muscles. This becomes important when you consider the idea of a motor unit, a motor nerve coming down, innervating a muscle. One nerve, when it branches, only innervates muscle fibers of the same type. So one motor nerve goes only to type 1s. One other motor nerve goes to type 2s. And it's in this way that we can organize recruitment of the entire muscle. Let me end with one other sentiment. If we were to do muscle biopsies on a lot of you, like you saw me in the video, and we teased out the different fibers, humans are marvelously adapted to have a really good even blend of each. So your soleus, your gastroc, your biceps, the diaphragm will have some slow and some fast fibers. Usually 45 to 55%. And some of them might be a bit more fast. Gastrocs are a bit more fast and more powerful. Soleus is a bit slower, but they're generally 50-ish%, half and half. Rats and mice, you'll see muscles that are 85% fast or slow. So motor units activating these fibers within a muscle activate different parts of the muscle. The more motor units, the more of the muscle you activate, the more of these fibers you recruit. So we'll leave it at that for today. We'll come back and talk about recruitment. We'll talk about tension and force generation, and then get into metabolism. Have a wonderful Tuesday.